Introduction. Cell death is a fundamental process that plays a crucial role in various biological processes, including development, homeostasis, and defense against pathogens. Understanding the different mechanisms of cell death is imperative for comprehending disease pathogenesis and developing therapeutic interventions. In this script, we will explore the intricacies of cell death, focusing on various signaling pathways and their dysregulation in certain diseases. We will discuss apoptosis signaling pathways, necroptosis, autophagy and cell death, pyroptosis, ferroptosis, death receptor signaling, mitochondrial apoptosis, executioner caspases, DNA damage response and apoptosis, metabolic regulation of cell death, crosstalk between cell death pathways, and diseases associated with dysregulated cell death. This comprehensive analysis will shed light on the importance of cell death in physiological and pathological contexts. Apoptosis is a highly regulated form of programmed cell death that occurs in response to various internal and external stimuli. It plays a critical role in tissue remodeling during development, elimination of damaged cells, and immune system regulation. Apoptosis can be triggered by two major signaling pathways: the extrinsic pathway, initiated by the binding of death ligands to death receptors, and the intrinsic pathway, involving mitochondrial dysfunction and release of pro-apoptotic factors. These pathways converge on a cascade of caspases, culminating in cell dismantling and removal without eliciting an inflammatory response. Dysregulation of apoptosis signaling pathways is implicated in numerous diseases, including cancer, neurodegenerative disorders, and autoimmune conditions. Necroptosis is an inflammatory form of programmed cell death that occurs in response to certain triggers when apoptosis is impaired or inhibited. It is characterized by the release of damage-associated molecular patterns, DAMPs, leading to the activation of pro-inflammatory signaling pathways. Necroptosis is mediated by the receptor-interacting protein kinase 1, RIP1, and RIP3, which form a necrosome complex that ultimately results in cell membrane rupture and release of cellular contents. Understanding the regulation and modulation of necroptosis is crucial for developing therapeutic strategies against a range of inflammatory diseases, such as ischemic injury and chronic liver diseases. Autophagy is a highly conserved cellular process responsible for the recycling of intracellular components to maintain cellular homeostasis. It acts as a cytoprotective mechanism under stress conditions, such as nutrient deprivation or organelle damage. However, under certain circumstances, excessive or dysregulated autophagy can lead to cell death. This process, termed autophagic cell death, involves the excessive degradation of cellular components and organelles, ultimately leading to cell demise. The intricate relationship between autophagy and cell death has important implications for various pathological conditions, including cancer, neurodegenerative diseases, and infections. Pyroptosis is a programmed cell death mechanism triggered by inflammatory caspases in response to infection by certain pathogens, including bacteria and viruses. It is accompanied by the release of pro-inflammatory cytokines, such as interleukin-1 beta, IL-1 beta, and interleukin-18, IL-18. Pyroptosis is distinct from apoptosis and necroptosis, as it involves the formation of gasderm and pores in the plasma membrane, leading to cell swelling and rupture. The activation of pyroptosis serves as an important defense mechanism against invading pathogens but can also contribute to the pathogenesis of infectious diseases and inflammatory disorders. Ferroptosis is a recently identified form of regulated cell death characterized by iron-dependent lipid peroxidation and oxidative damage. It is initiated by the depletion of glutathione and the accumulation of reactive oxygen species, ROS. Ferroptosis has been implicated in various pathologies, including neurodegenerative diseases, cancer, and ischemic organ injury. Understanding the regulatory pathways and molecular mechanisms of ferroptosis may uncover new therapeutic targets for diseases associated with oxidative stress and lipid metabolism dysregulation. Death receptor signaling is a critical pathway involved in initiating apoptosis in response to extracellular death ligands, such as FOS ligand, FOSL, and tumor necrosis factor alpha, TNF-alpha. The engagement of death ligands with their respective receptors triggers the recruitment of adapter proteins, leading to the activation of caspases and subsequent execution of apoptotic cell death. Dysregulation of death receptor signaling has been implicated in various diseases, including autoimmune disorders, cancer, and viral infections. Elucidating the molecular mechanisms underlying death receptor signaling may provide insights into targeted therapeutic interventions. Mitochondria play a pivotal role in regulating apoptosis by releasing pro-apoptotic factors, such as cytochrome C, into the cytosol. This triggers the formation of the apoptosome complex and subsequent activation of caspases. Mitochondrial apoptosis is tightly regulated by the BCL2 family of proteins, which control the permeability of the mitochondrial outer membrane. Dysfunctional regulation of mitochondrial apoptosis is associated with numerous pathological conditions, including cancer, neurodegenerative diseases, and cardiac disorders. Understanding the intricate mechanisms governing mitochondrial apoptosis is essential for developing targeted therapies against these diseases. Executioner caspases, such as caspase 3, -6, and -7, are central effectors of apoptosis. They are responsible for the proteolytic cleavage of critical cellular substrates, leading to the characteristic morphological and biochemical changes associated with apoptotic cell death. Executioner caspases are activated downstream of initiator caspases and play a crucial role in dismantling of the cell during apoptosis. Dysregulation of executioner caspases is implicated in various diseases, including cancer, neurodegenerative disorders, and autoimmune conditions. Understanding the regulation and functional roles of executioner caspases may pave the way for targeted therapeutic strategies. Maintenance of genomic integrity is essential for cell survival. The DNA damage response, DDR, safeguards cells against DNA lesions induced by endogenous and exogenous factors. In the presence of severe DNA damage, the DDR can trigger apoptosis as a protective mechanism to prevent the propagation of mutations. Dysregulation of the DDR and apoptosis signaling pathways can lead to genomic instability and the development of cancer. Understanding the crosstalk between DNA damage response and apoptosis may contribute to the development of novel cancer therapies and strategies to overcome chemotherapy resistance. Metabolism plays a crucial role in regulating cell death pathways. Perturbations in cellular metabolism, such as alterations in nutrient availability and energy status, can influence the sensitivity of cells to undergo apoptosis or other forms of regulated cell death. The metabolic reprogramming observed in cancer cells contributes to their evasion of cell death and promotes tumor progression. Unraveling the intricate relationship between metabolic reprogramming and cell death pathways holds promise for the development of metabolic targeted therapies against cancer and other metabolic diseases. Cell death pathways are interconnected and regulated through extensive crosstalk, enabling a coordinated response to various cellular stresses. Interactions between apoptosis, necroptosis, autophagy, and other forms of cell death influence the ultimate fate of the cell. The convergence and intersection of these pathways introduce complexity and fine-tuning to the cell death machinery. Dysregulation of crosstalk between cell death pathways can contribute to various diseases, including neurodegenerative disorders, inflammatory conditions, and cancer. Understanding the molecular mechanisms underlying this crosstalk may facilitate the development of targeted therapeutic interventions. Dysregulation of cell death pathways is implicated in the pathogenesis of numerous diseases. Cancer, for instance, often exhibits defects in apoptosis signaling and aberrant regulation of cell death pathways. Neurodegenerative diseases, such as Alzheimer's and Parkinson's, feature dysregulated cell death mechanisms contributing to neuronal loss. Autoimmune disorders, cardiovascular diseases, and infectious diseases also involve dysregulated cell death. Elucidating the molecular underpinnings of dysregulated cell death in these diseases opens avenues for the development of novel therapeutics targeting specific cell death pathways. Cell death is a complex and highly regulated biological process that is central to normal development, tissue homeostasis, and disease pathogenesis. Through this detailed script, we have explored various cell death pathways, including apoptosis, necroptosis, autophagy, pyroptosis, ferroptosis, and death receptor signaling. Additionally, we have discussed the role of mitochondrial apoptosis, executioner caspases, DNA damage response, metabolic regulation, crosstalk between cell death pathways, and diseases associated with dysregulated cell death. This comprehensive understanding of cell death mechanisms provides a foundation for future research and the development of targeted therapies aimed at modulating cell death in various pathological contexts.